Thank you very much. Thank you for the invitation to tell you today about the suppression of the latent reservoir as a possible approach for eradication. I think you're all familiar with this type of graph which depicts the viral circulating virus in plasma in an untreated infection, and as soon as ART is given to patients, there are drastic reduction in virus circulating in plasma to below the limit of detection. However, like Bob has mentioned, this is not the heart does not eradicate the provirus. A few cells remain latently infected that give rise to this rebound in viral replication as soon as ART is removed. Besides that, some patients show what is called blips, which are some episodes of viral replication that are detectable, and these blips are probably originated from reactivation of the viral reservoirs, even in the presence of ART. Some studies from the Silicanus group, as well as others, have shown that patients with have higher frequency of blips have higher reservoirs than patients with fewer blips. This kind of suggests that the reservoir is replenishment, and there's replenished, and there's some novel infections that occur even in the presence of ART that replenish the reservoir. So a logic approach for eradication would be to prevent this as much as possible viral replication and prevent these events of reactivation from latency. The idea is that this will lead to a progressive loss of the latently infected cells due to their natural turnover and over several years. If we think that HIV transcription could be considered as a therapeutic target, we see that there are no specific activators or suppressors of HIV LTR. Here is represented, it was deformed, but in the little squares you'll see all the binding factors for transcription factors. And as they are transcription factors that can be found also in cellular genes. So any approach that would promote activation or suppression mediated by these transcription factors would have an obvious effect on cellular activation. The only specific property really of HIV transcription is TAT and TAR. TAT is a transcriptional activator of HIV. In the absence of TAT, RNA polymerase starts transcription, transcribes approximately 59 nucleotides that will form this secondary structure called the TAR RNA, seen over there. But immediately RNA polymerase pauses because of presence of nucleosomes and negative elongation factors. Once TAT is recruited into the picture, TAT recruits a complex called PTFB, which is composed of CDK9 and cyclin T1. That allows the CDK9 to phosphorylate the C-terminal domain of RNA polymerase, as well as phosphorylation of the negative elongation factors and promote productive elongation. So TAT really is really important factor to regulate the passage from an active viral production to the entrance into latency. So the more TAT there is, the more transcribable active will be the virus, and as there is a loss of TAT, there will be also a silencing of the provirus. And TAT is really key to pass from the activation to this silenced phenotype. During this epigenetic silencing, there will be also accumulation of other epigenetic marks at the HIV promoter that will be recruited by histone deacetylases as well as transcription factors that will allow for a chromatinized promoter and prevent transcription from the HIV promoter. TAT has also been shown to be 300 times more efficient than any other cellular activation in terms of reactivation of the provirus. So these results would argue that TAT inhibitors would really have a very big impact in current therapeutic strategies by rendering TAT inefficient. By doing so, the provirus would enter this prolonged transcriptional silencing, preventing viral rebound and maintaining a permanent state of latency. So basically, the idea is that if we add a TAT inhibitor to current antiretroviral therapy, would be able to reduce this low-level ongoing viremia that exists even in the presence of ART to very low levels. We would probably reduce the size of the reservoir by preventing ongoing viral replication as well as any events of replenishment of the viral reservoir. And hopefully with time, the death of these long-lived cells would result in a continuous decay. So TAT is a really interesting protein. TAT is composed of 6 different domains. So far we only have a crystal structure of the activation domain, which is shown by the 3 first domains over there. It is being crystallized in combination with PTFP, which is composed of CDK9 and cyclin T1. And the basic domain shown in green is rich in arginines, and it's the region that binds to RNA, to the TAR RNA. There's some NMR structures of TAR, and as you can see, the green part in the NMR structures here on the right side seem to be very accessible. So TAT is a very excellent viral target because it's one of the first proteins to be expressed during infection. It has no cellular homologue. Like I mentioned, if we inhibit TAT, this blocks the feedback loop that drives the exponential viral production. And also, there's an added benefit is that TAT is responsible for a lot of the neurotoxic activity that is seen in AND, which is HIV-1 associated neurocognitive disorders, and TAT is a major player in those. So inhibiting TAT would have also that added benefit. So today I want to show a proof of concept, and this proof of concept of this suppression of the reservoir is based on it was basically because we have access to this compound called cortistatin A that I will tell you about today. So cortistatins are steroidal alkaloids that were isolated from a marine sponge called Corticium simplex that can be found in Southeast Asia. Initially, there was a lot of interest in cortistatin A because it shows some anti-proliferative activity against human umbilical vein endothelial cells at nanomolar concentrations. Unfortunately, this natural product is not very abundant in nature. You need approximately 1.5 kilograms of the dried sponge to yield 22 milligrams of cortistatin A. That sparked the interest of many synthetic chemists to synthesize this natural product, and Phil Barron at the Scripps Research Institute in La Jolla devised a strategy that departs from prednisone and requires certain chemical steps to synthesize gram quantities of DCA. The structure of DCA is over there on the left, the natural product, as well as an analog called the dihydrocortistatin A DCA. They're very similar structures. They have equipotent activity, and we have, in the next slides, I'll be talking about DCA, or the dihydrocortistatin A, because it was slightly easier to synthesize, and that's the analog we've been working with. So a while back, we showed that DCA is a very, very potent TAT inhibitor. At low multiplicity of infection, the EC50 is less than 1 picomolar, and at high MOIs, it's approximately 2 nanomolar. It binds to that basic domain of TAT and interrupts the TAT binding to TAR. So first, let's just give a couple of slides to show you that this interaction actually occurs and the mode of action of this compound. So we took advantage of a bioattenuated version of the compound, and you use that to perform pull downs using streptavidin bioattenuated beads. We expressed different forms of TAT, either the wild-type TAT, which is 101 amino acids, or a shorter version that is sometimes found in some isolates in culture, which is 86 amino acids, as well as a mutant of TAT that is mutated in the basic domain and therefore is not active in transcription. We transfected those in cells as well as some controls, namely this protein called 9G8, which is known to bind viral RNA. After the pull downs, we only see an interaction between bioattenuated DCA and TAT wild-type, but not TAT mutant or any of the controls. To show that this was a direct interaction, we performed the same experiments with recombinant protein. And as you can see on the bottom, if you incubate with increasing amounts of unbioattenuated cortistatin, you can inhibit this interaction, but you cannot inhibit the interaction with raltegravir. On the right side, you also have show that this bioattenuated DCA does not interact with the recombinant TAT mutant or a recombinant 9G8. Now, to show you that this is really interrupting the TAT binding to TAR, we perform RNA EMSAs. RNA EMSAs are based on, basically we take radio-labeled RNA and we incubate it with a protein. And if there is an interaction between the protein and the RNA, it will migrate slower in a gel. So here in blue, over on the top left, you'll have the radio-labeled TAR alone. If you incubate this with a basic domain peptide, so basically a domain of the TAT that is just covering the basic domain, you have an interaction with the RNA, and this will migrate slowly in the gel. Now, if you incubate with increasing concentrations of DCA, you will abolish this interaction. If you do the same experiment now, the central panel with raltegravir, you have no inhibition of the interaction. And if you use neomycin, which is a non-specific inhibitor of TAT and TAR, you can inhibit the interaction at very low concentrations. If you do the same experiments now in the middle left with a peptide that is targeting the C-terminus of TAT that does not interact with RNA, you don't see any interaction. We did the same experiments here on the bottom right with the full TAT, and you have in red you'll have a shift that is caused by TAT-TAR interaction. If you incubate with DCA, you inhibit this interaction, but that is not seen with raltegravir. So TAT, DCA has the ability to bind to the basic domain of TAT and interrupt TAT-TAR interaction. I want to show you here some properties of this compound. So if you perform an acute infection with this compound, this has a very different curve than any of the other antiretrovirals that are currently used in the clinic. The EC50 of DCA is really, really low. However, it does not achieve maximal concentrations during an acute infection. And this is mainly due because the transcription that is driven from TAT initially, from the initial transcription is mediated by a lot of cellular activity such as NF-κB and SP1. So thus DCA cannot inhibit that TAT-independent activity. However, if we take cells that are already chronically infected and they're all infected and we treat these cells with the TAT inhibitor, now we can reduce the HIV, the viral RNA that is produced from these cells, and the other retroviral compounds have no activity in this type of cells. So that is why this is a very interesting property of this compound. It's particular to this particular type of block of the viral life cycle, and that's why we decided to look at the effect of DCA in latently infected cells. So for that, we started out by testing DCA on a model that we created in-house, which was ELA CD4 cells latently infected with a provirus called PNL43. So we treated the cells, they were all chronically infected, so the same results could have been obtained with antiretroviral, but we treated them with the MSO as control in DCA, and over time what you see this reduction in P24 production, and we also confirmed that by real-time PCR there was approximately 99% reduction in viral RNA. So we see this is, we believe that at the time this was mainly due to transcriptional inhibition. So if that was the case, if we stop the treatment, if we remove the treatment, the promoter will be epigenetically repressed and there shouldn't be no viral rebound. So that's what we did here in green. We stopped treating the cells, and for the next 150 days we didn't observe any viral rebound. We performed some chromatin immunoprecipitation to confirm against RNA polymerase, to confirm that this was actually inhibition of RNA transcription. And as you can see on the left graph, RNA polymerase is recruited to the control cells in blue. However, if you treat with DCA, there's a drastic loss of RNA polymerase at the elongation of the HIV genome. And as a control, we use alpha-manitine, which blocks RNA polymerase. And as a positive control, activation of the latent cells with TNF, which increases the recruitment of RNA polymerase. On the right side, we didn't observe any effect in terms of elongation of any other genes, such as GAPTH. So now we wonder if this latency mediated by this compound could withstand a reactivation event. So we activated the cells with a combination of PMA and neomycin, which activates the PKC pathway, and we readily were able to activate the control cells, but we could not activate cells that had been that were in this DCA-mediated latency. To confirm that the pathways were still intact in these cells, that we did not select for cells that were deficient somehow in these pathways, we looked at the downstream activators of the PKC pathway. So we looked at phosphorylation of MEK and ERK, and as you can see in the top lane, with PMA ionomycin in either the latent cells or the DCA-mediated latent cells, you'll have an increase in phosphorylation as well of MEK and ERK. We also looked at IL-1 beta production. There is downstream effect of PKC activation, and this was similar between the two lines as well as an infected line. So now we wonder if we really did not select for some cells that somehow had lost the provirus. So for that, we decided to transfect large amounts of TAT to see if we could recover the virus from these cells. And that was actually the case. So in control cells, we were readily activated the virus. And in the deep latent cells, we were also able to reactivate the virus in the presence or absence of DCA. For our surprise, there was this return back to deep latent through this DCA-mediated latency. So the TAT that was produced was not enough to jumpstart the mechanism. So there's several possible explanations for that I don't want to go into, but this is just basically to prove that these cells still contain the virus just not actively transcribed. If we take the virus that was produced from these activations and we sequenced it, there was no deletions of this virus, and if you infect novel cells with this virus, it is fully competent for infection, and it's still inhibited by DCA. So we did the same type of studies with other cell lines that are more used by other labs, such as the promyelocytic cell line OM101, which contains a full-length provirus. And as you can see, in this case, we used ART or ART plus DCA at 2 different concentrations. And you see the same reduction in viral production over time. And that was confirmed by RT-PCR on the right side, showing that there was a drastic inhibition of viral RNA. In these cells, we can use different set of activators to reactivate the provirus from latency, such as SAHA, which is an HDAC inhibitor, or prostatin, a PKC activator, or cytokines such as TNF-alpha. And in all cases, we were able to show that the presence of DCA drastically inhibited this reactivation from latency. As a control, we tested a couple of cell lines that have certain peculiarities that help us prove that this was a TAR-dependent mechanism. ACH2 contains a provirus that has a mutation in TAR, and U1 cells contain 2 provirus. One doesn't have an ATG, and the other TAT contains a mutation that renders TAT activity suboptimal. So we use these 2 cell lines to prove that DCA was specific for TAT-TAR activity. And as you can see, if you culture these cells in the presence of DCA, there was absolutely no difference between the control and DCA-treated cells, and because there was not a competent TAT-TAR activity. If we now use HDAC inhibitors or PKC activators, you can see that the inhibition was very minimal in these cells. So now the important question was, would this work in CD4 T cells from infected individuals? So this was done in collaboration with Nicolas Chaumont and Lydie Trotman. And so we took CD4 T cells from patients, and the selection criteria for this patient was the following, is that when the CD4 cells were placed in culture, there was absolutely no detectable viremia, as you can see by not detected on each graph. And as soon as we used a strong activation, antigenic activation, such as antibodies anti-CD3, CD28, we would have a strong reactivation from latency. And so we performed these experiments in the presence or absence of DCA, and what we observe is that in all cases, DCA inhibited viral reactivation from as much as 100% to 55% is in subject C. In average, if we look at the graph on the right side, there was approximately 2 log reduction in terms of reactivation. So we next wondered what would happen if we treated the cells for a period of time, and then we were trying to simulate in vitro what would happen in vivo, this rebound after treatment cessation. So we tried to create a model where we took these CD4 T cells from viremic patients, we sort the CD4 T cells, we expand them lightly with PHA, IL-2, and feeder cells, and we culture them in media for 22 days in the presence of IRVs or IRVs and DCA. After that period, we wash the cells, and we ask several questions. What will happen if we maintain them in IRVs? What will happen if we stop the treatment? And what happened if we kept the treatment and activated them with prostatin? So here's the results from two patients. On the top, we have the experiment where we remove the treatment. So in the blue bars, you'll have the rebound that was observed in cell lines that were kept on IRVs alone. So if you stop the treatment for another 6 days, you'll have an immediate viral rebound. If you had prior treatment, now on the right side of each graph, if you had prior treated the cells with DCA and you remove all treatments, there's a very, very reduced viral rebound by more than 93%. Now on the bottom panel, you have the experiment where we kept all the drugs, but we activated the prostatin. And as before, in this period of 6 days, we've seen that there was actual no viral detected if the cells that were priorly treated with DCA and the activation was done in the presence of DCA. So since then, we've tried to optimize this assay because 6 days were not a very long period to test the absence of the drug. So we tried to optimize this assay, and we have now treated the cells for 28 days, and we followed the viral production for the next 21 days, 22 days. 21 days. So in the top, you'll have the viral expression 14 days after stopping the treatment, or 21 days later. So the first two graphs on left and right are pertaining to patient A, and the middle ones to patient B, and the bottom to another patient, patient C. As you can see on the top, we could not detect any viral production 21 days after no drugs whatsoever in the culture in patient A and in patient B, and very, very limited amounts in patient C. So, so far it seems that prior treatment with DCA can at least control viral rebound for the next 21 days. We looked at the viral reservoir, so we can argue that there's many ways to look at the reservoir. Here we just did an ALU PCR to look at integrated HIV DNA in these 3 patients. So this was the cells that were kept on IRVs or IRVs plus DCA for the period of 49 days. And what you can see is that in general there's a trend where we see a loss in proviral. So our hypothesis is that in culture, there's probably still ongoing some cell-to-cell transmission, and antiretrovirals that we used were not enough to control that cell-to-cell transmission. But in the presence of DCA, as there is no viral RNA produced at all, DCA can prevent that cell-to-cell transmission, and therefore with time we'll see this loss in proviral integration. So if cell-to-cell transmission is really something that is occurring in vivo, this kind of seems to indicate that treating with TAT inhibitor would have an impact on the size of the reservoir over time. So I hope this proof of concept was convincing enough to show to create this idea that maybe there is an alternative way to go about treating the reservoir, that maybe suppressing the reservoir is a viable alternative. There are several questions that remain that we're trying to address. One is that in terms of the mechanism of action, does this compound is simply accelerating a normal occurrence that is the typical latency, and what we would observe in terms of the HIV promoter would be the normal set of epigenetic modifications that you see in the latent provirus, or because we are only inhibiting a subset of TAT activities that are based on the basic region of TAT, this might result in unique epigenetic changes. For example, we know that TAT binds to SWIFT-SNF, to P300, CEBP via the basic domain. Other regions of TAT are involved in other factors such as PTFP, HCGN, etc., that are also involved in chromatin remodeling or transcriptional activity. So at the end of the day, by inhibiting only a subset of activities, you might end up with a different set of epigenetic changes that explains this lack of viral rebound. So in conclusion, DCA is a very potent inhibitor of TAT-activated transcription without cell-associated toxicity. Long-term treatment with DCA reduces low-level viral production to a state of true latency that is refractory to viral reactivation by latency reversal agents. Latent cell lines that have mutations in TAT or TAR seem to be insensitive to DCA, as expected. DCA inhibits reactivation upon a very strong antigenic stimulation of CD4 plus T cells isolated from virally suppressed patients. And if we prior treated cells from these patients with DCA, we seem to be able to suppress viral rebound for up to 21 days. And it seems like the content of the proviral DNA is reduced over time in the presence of DCA. So DCA defines it's a novel class of anti-HIV drugs that has this ability to decrease residual viremia during ART, and it should be considered as a promising drug to be included in therapeutic eradication strategies. And I would like to thank very especially to Lydie Trotman's group and Nicolas Chamon that did most of the work with the patient cells. A lot of people at Scripps have helped with synthesis of DCA, structural studies, PK studies, etc. We'll be studying the effect of this drug in humanized mice in collaboration with Victor Garcia at the University of North Carolina, and hopefully next year in non-human primates at the University of Wisconsin with David Evans. And of course, this is my group that I have to thank a lot. Most of the work I showed here were performed by Guillaume, by Carrie, and Sonia. Thank you very much, and take any questions.